Hello everyone, and welcome to phyloseminar.org, a project supported by the Society of Systematic Biologists. For people with even a passing understanding of molecular evolution, it should go without saying that there are different substitution rates across sites. In 1994, Zhiheng Yang made a substantial advance in modeling rates across sites by using mixture distribution based on categories of gamma-distributed rates. This early paper has made it squarely into the canon of phylogenetic analysis. Although this simple model has been useful, real sequence evolution deviates significantly from this model both between sites and across trees. For the series of 3 talks, we are hearing from 3 individuals who have thought a lot about modeling heterogeneity in the substitution process across sites. Today's first talk will be from Andrew Roger, who will motivate the need for more advanced models with evidence from deep phylogenetics and introduce the cat models. The second talk will be from Nathan Whelan, who will propose a site partitioning approach to handle the same issues. Finally, we'll hear from Nicolas Lartillot, who will describe Dirichlet process mixture models for site heterogeneity in detail. As I mentioned, today's speaker is Andrew Roger, who has consistently been a thought leader in deep phylogenetic relationships and methods to infer them. Andrew has studied phylogenetics throughout his career, starting with a PhD advised by Ford Doolittle in the 90s. After that, he did a postdoc with Mitch Shogan at Woods Hole. Then he joined the faculty at Dalhousie, where he's been a full professor since 2009 and holds a Canada Research Chair. Welcome, Andrew, and thanks for participating.
Thanks very much, Eric. As Eric mentioned, I guess we're going to focus on phylogenetic modeling and the kinds of problems that you encounter in phylogenetic modeling when you have very deep phylogenetic divergences, such as the deepest divergences in the tree of life. And that's where I come from in all of this, is trying to understand what the deepest branching groups are in the eukaryotic tree of life. So here's a depiction of our current view of what the deep eukaryotic tree of life might look like. And these are major groups, just to orient you. Animals are right here, plants and fungi are over here. So the majority of eukaryotic diversity is not encompasses within those so-called kingdoms, but instead in the microbial realm, and that's what's shown in most of these lineages are major supergroup names that we and others have proposed for these major groups. The difficulty with this eukaryotic tree of life is that most of the interesting deepest branches are very, very old. And in fact, when it gets right down to it, and we use molecular phylogenetics, it's actually quite difficult to reconstruct anything in this zone here, where the question mark's indicated, which probably goes back somewhere between 1.2 and 1.7 billion years ago. And so the reasons for that are several-fold. I'll just show you first a concatenated protein phylogeny based on about 150 different protein markers. Matt Brown made this phylogeny using the CAT-GTR model in PhyloBayes. And you can see that the major supergroups of eukaryotes on the right in the different colors. And what I want to draw your attention to is the extremely short internal branch lengths near the base of this phylogeny. And that's part of the difficulty with dealing with deep phylogeny, is that you often have very great differences in the rate of evolution of different lineages, but also you potentially have rapid radiations deep in the tree, and those rapid radiations mean there's short internal branches that are very difficult to reconstruct. So in general, if we were to say what were the problems that we encounter and most people are encountering in deep phylogenetic reconstruction, I think in the 90s and the early 2000s, we were in the situation of not enough information. That is, people were using single or a few genes to try to reconstruct deep phylogenetic problems, but really there was not enough data for those single genes or proteins or small sets of proteins to get at the deeper branches, those short internal branches. And so what has happened with the rise of next-gen sequencing is the approach of increasing sampling of multiple different genes from many different taxa and using these genes either through concatenation into giant super matrices of alignments or taking individual gene trees and somehow synthesizing the information from individual gene trees using supertree type methods. The problem that comes with these deep divergences, and with any divergence in fact, is that we're always trying to struggle with the fact that we're estimating a species tree, but the underlying molecules we're looking at are actually evolving along gene trees. And so that leads to problems that have been discussed in this Phylobabble series, such as incomplete lineage sorting, where the multiple alleles in ancestral populations differentially sort as speciation events occur. So the actual gene tree is not the same as the species tree. Or cases where you have events of gene duplication and differential loss that can actually mask horology, ancient gene duplication kind of mirror trees, and make us think that we're looking at orthologs when we're in fact not looking at orthologs. And then in the problem of lateral gene transfer is particularly problematic in microbial evolution, and that is where genes move between organisms. And to the extent that lateral gene transfer occurs, it's very difficult to actually reconstruct a tree of life knowing for sure that the genes are all singing the same song and came from the same history. But today I really want to focus more on a third source of problems, and that is difficulties in modeling the actual process of sequence evolution. And these difficulties not just stemming from lack of information, but actual misspecification of models that don't properly take account of the evolutionary process. And in many cases, such model oversimplifications can lead to systematic error. And that can often take various forms, but the most commonly encountered problem is the so-called long branch attraction problem. I will be talking a little bit about another problem that's related called long branch repels. So our current models of sequence evolution really owe a lot to the pioneering work of Margaret Dayhoff in the 70s and 60s. She actually introduced some of the first protein models of evolution, and some of these models we still use today. Often people suggest that we should be modeling protein evolution on the level of the codon, that is the codons that specify the amino acids in the sequence. And this is probably the best way of going about modeling things, but it can often be very difficult. For instance, if we are looking at the codon models naturally have 61 by 61 matrix of interchange between codons, and that's very complex type model to work with and leads to computational difficulties. There's other problems with codon models that I won't discuss here, but I'm going to devote most of my talk to actually amino acid level analyses. And this is where Margaret Dayhoff's famous PAM matrix came in, where she assembled from databases of sequences, looked at amino acid changes, and developed rate matrices, essentially, of the rate at which one amino acid would transition into another in a particular, any particular protein. So these overall single matrix rate matrices, such as the PAM matrix, were very important and are still used today. So the newer models that we've been developing in the last few years, such as the LG, JTT, WAG, PMB, and mitochondrial specific models, these are all models that are based on databases of amino acid changes. They're differently estimated in the case of these different models, but they're all once and for all type single matrix protein models. So these protein models are often combined with some kind of rates across sites distribution. As Eric alluded to, Ziheng Yang brought in the use of mixture models in case of applying a discretized version of the gamma distribution to different rates at different sites and proteins. So the LG plus gamma or LG plus variable sites models are there to deal with different rates at different sites. And then finally, the matrix is often adjusted to the equilibrium frequencies of the amino acids of the data set. So some proteins would be rich in some amino acids, and so we often can adjust the LG matrix to have a different stationary distribution by using the equilibrium frequencies or the observed frequencies of the alignment that's under analysis. So that's the LG plus F plus gamma model, for example. So what I will talk about now is a kind of basically following up on the idea of concatenation and taking individual protein genes, putting them together from the same species and trying to estimate phylogenetic trees and the problems that come with that. So if we to look at this sort of schematic of a multiple alignment and just focus in on a segment of particular protein G, the amino acid sequences from the various taxa are shown there. And I've made those up completely, by the way. Those don't correspond to any real protein as far as I'm aware. And what we want to do is evaluate, with likelihood-based methods, we want to evaluate the probability of observing the sequence at the tips of a phylogenetic tree. So in this particular site X, we have I, F, I, and V in Lactobacillus E. coli, human, and shiitake mushroom, respectively. So this is the problem, the phylogenetic problem, is to understand what is the probability of observing this data given this topology. And then for maximum likelihood type approaches, we will try and sum the log likelihoods, site log likelihoods over all sites, and then find the tree with the global best topology in terms of its maximizing the likelihood of the observed data. Bayesian approaches have the similar likelihood function at their core, and that's what I'll be discussing, the actual process model for sequence change. So really, the heart of the calculation is this probability of going from a given amino acid state I to an amino acid state J for a given protein G in your supermatrix. And of course, we have to look at it on a site-wise basis. And so the site likelihoods are calculated. And then we are, in fact, going to drill down instead of the site likelihoods, we're going to go right down to the branch to see how the actual model influences the probability for a given branch in the tree of going from state I to state J in a given period of time. So just shown in more precise form, this is the probability of being in state J, starting the branch in state I over a given length of branch. We're going to call it length of T. So this is really the core of the single matrix type model approaches for phylogenetic estimation. And it's this matrix of probabilities of transition is given by the exponential of a rate matrix, and it's also the rate matrix is scaled by the branch length and the rate of evolution of the site. So we'll go through these one by one. The rate matrix here is just the relative exchangeabilities of different amino acids. And that just suggests how quickly one amino acid could evolve into another relative to other amino acids. And it's shown here on this bubble plot. Large bubbles mean more rapid rate of interchange. So that actually gives some information about what amino acids are more likely to substitute for each other over a given period of time. But there's also this second feature that I discussed, the equilibrium frequencies of the data set, and that's given here by this capital Pi. This is a diagonal matrix of frequencies, amino acid frequencies from A through to V. Here are the diagonal entries in this diagonal matrix. And this is the stationary distribution of the matrix. So this is, if given enough evolutionary time, you ought to see amino acids occurring with frequencies shown in this diagonal matrix. If, say, the branch lengths get long enough, or you look at an alignment that's evolving at a certain rate, if you look at enough taxa, you should ultimately get a stationary frequency distribution of amino acids at that site that are basically this diagonal matrix. Of course, the rate of evolution for a particular branch is allowed to change and then allowed to be different amongst branches. So here at branch E, I've shown that by this t, this value of t. And so that is optimized during maximum likelihood. The optimal t for every branch is estimated. And again, it works into these matrix calculations through this multiplication. And then there's the rate of evolution at the site. So this is accounting for the fact that different sites have different levels of constraint. And so some sites may be highly conserved and not change at all. Others change very rapidly. And so you could have different rates, intrinsic rates. And this basically is just a multiplier of the branch length, making fast rates go faster and have longer branch lengths than slow rates. So the way that this is handled, and I'm not going to discuss it in detail, is not that it's actually dealt with by estimating a rate for a given site, but it's actually dealt with in the mixture model format that Eric referred to earlier. So we're going to average over different instances of this R coming from the gamma distribution. And we'll do that averaging on the level of the site. Different site likelihoods will be calculated under different R's coming from the gamma distribution. And then it's averaged by the probability of those rates. So that is our single matrix model, and that still today is probably the most widely used model of amino acid sequence evolution. And that's it's done incredibly well over the years. The difficulty is, and this comes actually from an earlier Phylobabble talk, so I'm stealing this from Richard Goldstein, that really, unfortunately, it's a spherical cow type model of protein evolution. So by that I mean it's an abstraction. It's extremely simplified, oversimplified in many ways, and it's done in so as to make the calculation feasible, and that was the original reason for that. The problem is it's nothing like how real proteins evolve, and it's the averaging over many sites, different types of sites, different lineages, has actually given rise to a widely and broadly useful model, but a model that actually has some serious shortcomings, especially when you're looking at the deepest branches in the tree of life. So how can this model be wrong? And I'm going to go through some specific ways in which this model is known to not adequately capture the process of protein evolution. I refer people to the other Phylobabble talks that have dealt with this issue previously. There's a number of really interesting ones. Richard Goldstein is the one that deals specifically with some of the issues of protein structure that I'm going to discuss here today. So how about the assumption of these models? Well, let's just start with parts of the models divided up. We'll look at the stationary frequencies of amino acids. So the assumption of this spherical cow model is now that we have a stationary frequency distribution. That is, that the amino acids occur at particular frequencies. Unfortunately, that means that they're supposed to occur at those frequencies in all species at all sites, and that they do not change over the process of evolution. This is clearly not true. If we actually just look at the amino acid composition of different proteomes. So this is the overall amino acid composition of proteins encoded in a given prokaryotic genome. Each dot here represents that. And what is depicted here is the GC content of that genome in the third codon position. So high GC is up here at 100%, low GC is down here near 0%. And on the y-axis is shown amino acids grouped according to whether or not they have G and C in the first two codon positions, or A and T in the first two codon positions. So GARP is GC rich, FINKY is AT rich. And as you can see, there's a very clear correlation between high GC organisms with a lot of G and C in their genome, and having a very high composition of the G, A, R, and P amino acids. Vice versa, very low GC organisms tend to have enrichment in the Fi, Nk, and Y amino acids. So, and the range here of variation is actually quite a lot. So that's going to affect individual proteins that we are making phylogenies of. And it's something that these single matrix models do not accommodate at all. The expectation is a single stationary distribution. Okay, so here's another assumption of these models, that the rate of evolution at a site is constant. So fast evolving sites are always fast, slow are always slow. And so, and on different branches of the tree, a given site will have a given sort of clock speed. This is actually demonstrably untrue as well, especially if you look at the deeper parts of the tree of life. Here's a segment of the elongation factor 1 alpha protein, the protein that delivers the tRNA to the ribosome, the charged tRNA to the ribosome during translation. And it's just an aligned region between archaea or archaebacteria versions versus the eukaryotic version of the protein. And if you can look at this, what you immediately see, if you look closely, is that at certain aligned positions, certain sites are evolving quickly within archaea and are completely invariable, or invariant at least, within the eukaryotes under examination. And I've chosen the phylogenetic breadth of the archaea and the eukaryotes here to be roughly equal. So this is clearly a case where there are different rates in different parts of the tree at a given site. And in fact, if you look more closely, you can see similar kinds of things going on. You can also see another pattern where, in fact, the type of amino acids allowed at sites is changing between eukaryotes and archaea. And our single matrix spherical cow model does not account for this. It will not allow you to have changing rates of evolution over the tree. Here's another assumption. One is that if we're looking at concatenated gene phylogenies, that if we look at different proteins, so different proteins over your concatenate, that for a given branch length, they should all have the same branch length, same degree of divergence for that particular branch. Well, this is also not true. And I can demonstrate this simply if we just take a little cartoon of a concatenated alignment, and I've actually extracted from the concatenated alignment two real genes, an alpha-tubulin gene and a heat shock protein 90, and these are eukaryotic microbe sequences and phylogenies inferred from each of those sets of organisms. And what I want to draw your attention to is the fact that the branch lengths leading to the taxa are actually quite different in the two genes. It's not just a matter of scaling. One is a more fast evolving gene than the other. The relative branch lengths are quite different. So here paramecium is going slower than cryptosporidium, and in this gene it's vice versa, that paramecium is going faster than cryptosporidium. And here the overall divergence is quite a bit more than the overall divergence. So different genes have different branch lengths or other model parameters for different proteins. So that is another thing, that if we use a single matrix model for our concatenated alignment, we're not going to be able to accommodate. Finally, I'm going to get to the part that I really want to focus on, which is that the assumption that the actual model of sequence change, the relative rate of amino acid substitution, the so-called rate matrix of change. Here I've got R times capital Pi, which I will be referring to together. The product of these matrices is a Q matrix. And the assumption here is that different sites in proteins and in your concatenated alignment are all playing by the same rules. They're all evolving according to the same kinds of forces, those forces reflected in the single matrix model. And there's also the assumption that the different sites in the molecules are independently evolving of each other. So as for the fact that all sites are playing by the same rules, well, any biochemist can really tell you that in a protein structure, that is clearly not the case. Here's just a 3D structure of the backbone of lobster enolase, showing the active site with some of the side chains there. Now, if we map onto that backbone all of the location of the hydrophobic amino acids, you should see that on the right. You get this kind of surface rendering which shows that they're towards the core of the enolase protein. But if we now ask, what about acidic and basic amino acids? Well, they're actually much more often near the surface. So you can see between these two that not all sites here in this molecule are likely to be playing by the same rules. And it's well known that surface residues tend to be polar or acidic and basic. Core residues tend to be hydrophobic. And that's going to affect how these molecules evolve. The sites on the surface are not going to evolve by the same rules as the sites in the core of the protein. Furthermore, sites are not independent at all. And shown here is a diagram of the GROE-L, GROE-S chaperonin from bacteria, a giant molecular machine in prokaryotes and in our mitochondria that refolds proteins. And it's a very large multi-subunit protein that I'm just trying to highlight here an analysis done by Mario Fares' group, which shows that particular residues within these molecules shown here are actually co-evolving. So they can be shown to be statistically dependent on each other. So the identity of one residue affects the identity of the other. And there's, in fact, a very large literature in the coevolutionary field to understand how different parts of proteins can co-evolve and affect each other. And it's actually quite useful to look at coevolution as a way of predicting structure, because things that tend to interact with each other physically will often tend to co-evolve. So that's, again, a violation of our single matrix model, which assumes that all sites evolve independently of one another. So really, what is the project here? And I think the project has been for quite a number of years, is to go from this kind of spherical cow model, a model of evolving spherical cows here. Now these, just think of these as our abstracted single matrix protein model as applied to proteins, drawn as small spherical cows, or actually circular cows in this case. And this is the model that we've been working with for many years. But maybe what we should be doing is, in fact, trying to improve this model and actually get a more realistic model that actually looks at how proteins truly evolve. So I'd say the overall goal of our work and many others in this field is to try to figure out if we can capture enough of the true underlying process of protein evolution to allow more accurate phylogenetic reconstruction at the deepest level. But one problem is that you cannot possibly model the process exactly realistically. You end up with far too many parameters. You get into this infinitely many parameters trap that statisticians discuss. And so the goal really is to actually see which parts of the process are really the most important to phylogenetic estimation, which parts can we abstract and leave a little bit more spherical cow. if that's an adjective. So there has been a lot of progress in this area, and I'm not going to summarize it all. It's summarized on this slide in case anyone wants to go back and look up some software tools that they can use for various problems that they've encountered. But the changing amino acid composition has been, to some extent, been addressed by people modeling in a non-homogeneous model, that is where the frequency stationary distribution is changing over the phylogenetic tree. And these are some software tools that allow you to try to do that in protein evolution. The issue of heterotachy, the fact that different rates occur at different parts of the tree at a given site, has been dealt with in the context of so-called covarion type models in these software tools. And recently, IQ-Tree, the creators of IQ-Tree, have come up with a mixture of branch lengths model, which also deals with heterotachy in a distinctly different way. Site interdependence is actually one of those problems that is not going to be easily dealt with, because once you start considering sites as being dependent on each other, evaluating the likelihood of an entire alignment becomes extremely complex. There have been many studies now trying to deal with site interdependent structure-informed models. I encourage people to go and look at work by Jeff Thorne, Nick Rodrigue, Nicolas Lartiot, and a number of others, including Richard Goldstein, who've really thought heavily about this site interdependence problem. But at this moment, phylogenetic models cannot be really used for large concatenates using site interdependence. And then there are ways of dealing with this issue of different genes having different rates of evolution for different lineages. The so-called partition models that are now widely implemented in different software packages, which allow you to partition up your data matrix into individual proteins. Or again, this mixture of branch lengths model might actually also accomplish the same feat. What I wanted to get to really is this issue of site heterogeneity in the amino acid substitution process. And that's what I'll spend the rest of the talk dealing with. And that rest of this talk is really combined work that I've been doing with Dr. Huaiqin Wang, Ed Susko, and collaboration with Dr. Bui Quang Min, who's in Vienna, and he's the principal developer of the IQ-Tree program. So let's go back to this idea of site-specific constraints in evolution. This is a segment of the chaperone 60 alignment. This is taken over from plants, fungi, animals, from our mitochondrial protein, and then the bacterial homologs. And I want to draw your attention to some particular sites in here. And these sites are instructive because they really deviate from what you would expect from a single matrix model. So if we look at this site, for example, it only has V and L, or this site over here only has R and K, and maybe in one case we have an A there, DRE over at this site. Okay, and we're talking about 1.5 billion years worth of evolutionary divergence captured here. So if I were to bet, I would bet that no matter what organism you look at, you're not going to find an arginine, for example, at this position where you only find D and E. There are really strong constraints in this position to be an acidic residue. So that's a chaperone 60 protein. And what's different here than what we expect from the single matrix model is that we don't, with the single matrix model, given enough evolutionary time, every site should eventually settle down to the same frequency distribution of amino acids. You should get all amino acids occurring with a certain frequency at a given site, given enough evolutionary time. And that doesn't seem to be the case. So we did a simulation based on the HSV90 protein, where we simulated under a single matrix model, a gamma distribution, and an overall frequency. We looked at the distribution of the number of different amino acids you would see at a given site in that alignment. And that's shown in purple. In light kind of mauve color, there's the HSP90 protein itself. And what I want to draw your attention to is the fact that under the JTT gamma plus F model, you actually see a smoother distribution of amino acids observed at sites. And you see a longer tail, more sites with more amino acids, and fewer sites with only a few amino acids relative to real data. So this bump here is this excess of conserved sites where the rules of what your amino acid identity is are constrained. And those rules you don't see coming out of a single matrix model. And you can actually quantify this over multiple different proteins, look at the uniformity of amino acid distributions and the number of states at a site. And that is what is shown here in this big table for 21 different protein families that we looked at. And I don't want to belabor the various tests that you can go and look in this paper if you want to see what we did. But the key here is that we're comparing the simulation prediction to the real data and then asked, are they significantly different? And what you can find is for every protein, given these different tests, you can at least find several columns which are significantly different with a p-value of less than 0.001. So real data does deviate from simulated data under single matrix models. It's significantly less uniform, and you tend to see fewer states at sites in real data than you would predict under a single matrix model. So these 2 points are what I just said. Real sites and proteins tend to be more restricted in what they can evolve to. And the real issue with this is it doesn't really matter unless it affects phylogenetic reconstruction. And the issue is that it actually does. And it was shown by Halpern and Bruno in 1998 that single matrix models underestimate the number of substitutions that occur along branches. And the underestimation gets worse at greater distances. So that causes a problem for phylogenetic estimation. Specifically, again, this commonly occurring problem of long branch attraction, where if this is a true tree of 4 taxa, and we let these branches A get very large relative to B, and B stays quite small, if you do not account for site heterogeneity and these site-specific constraints, as A gets very large, you'll start to see this long branch attraction. That is, the phylogenetic method will estimate the long branch attracts tree. And that has been shown by a number of different workers, given these references here. I'm going to just show you what we get under simulation. So here I simulated data under site-specific frequencies observed in an HSP90 alignment. So the frequencies of amino acids observed at sites to parameterize a model. So the frequencies for every site are coming from the observed frequencies at that site, plus the JTT exchangeabilities and a gamma distribution. That's how the data was evolved. And then what we did was we asked how often, under different settings of A and B branch lengths, could we get the right tree. And so shown down here is a heat map. As A gets very large and B gets very small, the frequency of getting the right tree in this region of the diagram, each one of these pixels is 100 simulations, and the color is how often we got it right. If we got it right, it should be white. If we got it wrong, it's black. You can see that there's a clear long branch attraction problem up in this part when we simulate under site-specific frequencies. So this region here is so-called Felsenstein zone of long branch attraction. And this is under maximum likelihood using a single matrix model, ignoring the fact that there are site-specific frequency constraints. So that's the phylogenetic problem. So, of course, there have been ways of dealing with this, and that's really been quite an odyssey, and many different people have been trying to deal with this. Rather than using these single matrix models, where all sites in an alignment are evaluated in light of one set of exchangeabilities and one set of stationary frequencies, we've actually moved to trying to get cases where we can have multiple rate matrices for different sites or different types of sites, if we can classify sites into different groups to try and capture this heterogeneity. And that's been done in a number of different ways. Halpern and Bruno did it in a way where they actually parameterized a codon model with amino acid frequencies estimated from every site. And this was a valiant attempt. Unfortunately, it showed some serious problems. That is, as you add sites, you're also adding 19 parameters for every site. That is, the stationary amino acid frequencies are parameters. So you're never getting more and more data to estimate the same number of parameters. And it has this infinitely many parameters type problem. The other problem is that there's a bias in the estimation that the unobserved amino acids will always be estimated as having a frequency of 0. And that's a real problem. And there's a paper by Nick Rodrigue in Genetics in 2013, and I meant to put the reference in there, but I guess that can be homework. Anyway, so that's the Halpern and Bruno approach. There've also been dozens and dozens of papers where people try to use information from the 3D structure of proteins to actually inform what kind of amino acids should occur at a given site. The difficulty with such models is that you actually need 3D structural information in order to make these models. They also tend to be computationally intensive. And for large concatenated alignments, you're not going to get that kind of information, at least not yet. Jesse Bloom has come up with a mutational scanning method, and he actually gave a phylobabble talk earlier, so I'm not going to talk anything about that. And I'm going to focus instead on mixture models. So what are mixture models? Well, there's two flavors really that have come up in the literature. The first was developed by Nicolas Lartiot and Hervé Philippe in a Bayesian paradigm, so-called CAT mixture model. And this is a model where they actually allow potentially an infinite number of different site types. That is, there's K different classes, and these classes each have a site-specific frequency distribution and one general exchangeability matrix. Okay, so this exchangeability matrix can either be uniform, such as the Cat-Poisson model, or you could make it more complicated, general type, time reversible type model that has 189 exchangeability parameters. And the dimension of this model actually changes during the MCMC that's conducted, and that the number of classes is controlled by a Dirichlet process prior. This is implemented in the Phylobase package, and it's actually become one of the most widely used models for phylogenomic analysis of big supermatrices. Nicolas Lartiot will talk about this in his talk. The alternative to the Bayesian approach is maximum likelihood implementations of mixture models, and that's been done by many different workers as well. And the approach here has been more to try and estimate a finite number of site types or site classes using frequency profiles again, often, where we look at databases of proteins and actually try and figure out, are there a finite number of classes that cover different types of sites in our own data? And then in a mixture modeling paradigm, we fit weights of the mixture to our data set using maximum likelihood. So the well-known models here are things like the CF4 model, LG4X model, which is actually 4 different rate matrices mixed in with rates of evolution. So this is a unique type approach. And then Le and Gasquiol and Lartiot came up with the C-series model, where you have the mixtures of 10 classes up to 60 classes based on a database of site types. And these are now frequency profiles that you will tend to see in the database, and that you can now fit to your own data. So I will now just briefly go through the mixture modeling approach. The theoretical justification for this is really beyond my expertise, and there is quite a lot of literature out there for the mixture modeling approach. But I'll just show you how it's done, and then discuss some of the approximations that we've come up with. So this is just the CF4 model to give you an idea of what I'm talking about. And these CF4 model has 4 classes of sites, and each of these classes represent here by a frequency histogram, and the heights of the bars indicate the frequency of the amino acid in that class. So these are basically saying there are sites in the data that have a lot of I and L and V. There's sites in the data that have a lot of G and some A. There's sites in the data that are very focused on D and E. And then there's sites in the data that have a more uniform distribution. And that's the so-called CF4 model, one of the simpler mixture models. But the C10 to C20 models, C60, sorry, models are very similar to these, but have a much richer set of different site profiles to try and capture real protein evolution, where constraints may not just fall into 4 classes, but you may see a much wider range of patterns. Okay, so site profile mixture models are basically, the idea here is that you have multiple Q matrices. So you have a general exchangeability matrix, and then the different site types are modeled by the different stationary distributions, that is, different site frequency profiles. Here I have capital Pi C. So a given class QC is given in the model by the product of the exchangeability matrix, which is general across all classes usually, and then a particular class frequency diagonal matrix. Now with mixture modeling, the issue is that we don't know which site comes from which class. And so in the absence of that knowledge, what we do is make a weighted average of the site likelihood over all the different classes. So that's given here. So if this is the probability of the data, given the tree and a particular class, rate matrix class, we can actually get the joint probability of that site and the class by multiplying through by the probability of the class itself, the frequency with which that class occurs in the data set. And that's given by these weights. So the mixture model approach occurs at the evaluation of site likelihoods. We evaluate a site likelihood under a given Q matrix, and then we do that again for another Q matrix, all of the different Q matrices that are in our model, and we then combine those as the weighted sum of this joint probability of the data and the rate matrix. And so that's briefly how the mixture modeling approach works. This is exactly analogous to how mixture modeling occurs with the gamma distribution for rates, except in that case, instead of Q here, we'd have the rate coming from the gamma distribution, and the W would be the probability of that class under the discretized gamma distribution. So that's basically how a mixture model works. In the Bayesian context, we might have different, in the, say, the cat model, this K is actually something that can change during the analysis, and there's all the prior probabilities associated with how many classes there are, and this weight here is also a prior probability. In the maximum likelihood approach, this weight can actually be optimized based on your data. So you get the overall likelihood of your data for a given set of weights, and then you re-optimize these weights to try to maximize the likelihood of the whole data. So that's the mixture modeling approach, and by and large it's worked very well. So you asked, what is the problem? Why can't we just use mixture models that have been introduced? What has happened is that the number of genes and the number of taxa available from diverse corners of the tree of life has increased dramatically with next generation sequencing. And as we try to get more signal and get larger and larger concatenated data matrices, these mixture models become computationally infeasible. And just to give you an idea, for a recent maximum likelihood analysis we did with IQ-tree using this mixture model, the C60+F model with an exchangeability using the LG model as the exchangeabilities and a gamma distribution with 104 taxa, 90,000 sites in our large concatenated matrix. The model took more than 350 gigabytes of RAM to run and about 3 weeks on 12 computer cores just to estimate the optimal tree using IQ-tree. And those of you who've used IQ-tree know that it's extremely fast. So this is not an implementation problem. This is inherent to mixture modeling in the maximum likelihood context. And because of that, there's no way we could possibly do bootstrap analysis. We can just get optimal trees. Similarly, if you use the Bayesian approach using the MPI implementation, the parallel processing implementation, it takes weeks to get thousands of MCMC generations, not nearly enough to get convergence. And even after running it for as long as you can, the problem is that we often in these settings never get the different multiple MCMC chains to converge on the same posterior distribution of trees. So we're just running into a wall here where convergence problems with phylobays and computational complexity for maximum likelihood are making it impossible for us to do this. So we really need faster methods. So I'm going to, in the time that remains, and I don't have a lot of time, I realize, but bear with me because I think this is worth it. We've recently been coming up with approximate methods to try to speed up this problem and make it actually feasible to do the kind of site heterogeneous analyses that are so important for getting accurate phylogenetic trees. So I'm going to introduce to you what we call the posterior mean site frequency approximation. And it's actually pretty simple for those of you who are used to mixture models. For those of you who are not, I'll just walk you through it briefly. The reason that the mixture models take so long to evaluate for maximum likelihood is that for every kind of rate matrix you're going to look at, every class of site, you have to evaluate the site likelihood for that class. So C60+F means you have to evaluate 61 different rate matrices for a given site. And that's going to take approximately 61 times longer than a single rate matrix analysis to do. And that's why it's so computationally intensive, and that's not even counting the additional overhead of parameter estimation. So rather than evaluating the mixture at every site, what we're going to do is approximate the model at a site, similar to how Halpern and Bruno did. They try to estimate the frequencies at every site, but that ran into problems. We're not going to estimate them in the same way. We're going to approximate them using a posterior mean estimate. Okay, and this is essentially an empirical Bayes type method. So what I've shown here in this equation is at a given site, we can find the posterior probability of a given class C, given the data. If we go back and now we look at the probability of that class C times the likelihood of the data given that class C divided by the total site likelihood. So the fraction of the total site likelihood that is the probability given by this class. And that'll give us the posterior probability of a given class at a site. What is the most probable class at a site? Now that is quite a discrete thing. Say if you have 20 classes, it'll be one of 20 things. But the key to our approach here is that instead of taking the most probable class, we try to average over different classes weighted by their posterior probability. So we take the frequency at a given site, X, we're going to, of amino acid J, we're going to take that frequency for the given class and then multiply that by this posterior probability of being in that class at the site. And this gives us a posterior mean estimate of the frequency of an amino acid at a site. So what this means is that we can actually develop a site-specific model based on the posterior mean frequencies of amino acids coming out of a fitted mixture model. So the first step in our procedure is to take a guide tree, say your single matrix tree, Lg plus F plus gamma, fit the mixture, fit your mixture model, your favorite mixture model on that single tree, and from that single fitting, you can actually estimate these posterior mean frequencies, and they now become the model for every site. So that is basically how it works. So now every site has its own model parameterized by its own site profile that we get by this posterior mean frequency vector. So by doing that, we no longer have to evaluate the likelihood k times for k numbers of classes. And so if we just look at the CPU times of these PMSF models, this is on a data set of 43,000 sites and 68 taxa, you can see that the CPU time for a given model for this large data set for the single matrix model is 43 hours. For the mixture models, it's quite a bit more, roughly 20 and 60 times more. And then these PMSF models that we've just introduced are maybe 2 to 3 times, in this case is less than 2 times, but often it's 2 to 3 times slower than the single matrix model, but much, much faster than the mixture models. And the amount of RAM that's required is barely more than the original single matrix model. So then the question is whether these models actually work as well as a mixture model. So we tried a number of different simulations to evaluate this, and we're going to use those same grids of simulation conditions where we have the so-called bunny ears tree with the long branches and the short branches, that when A gets large and B gets small, you get the long branch attraction bias. We're also going to look at a case where the correct tree has the long branches together, and sometimes models will actually push the long branches apart and estimate a long branch for Pell's tree. In this case, there's 2 different alternative long branch for Pell's tree for a given correct tree. So we're going to evaluate these new methods in light of these 2 shapes of trees to see how they perform. So here's simulating the data under a standard mixture model, so 21 different classes of sites. And then we're trying to recover the tree from the data. Each box in these squares here represents a particular branch length setting. So here it would be branch length A of 1 and branch length B of 0.02. So a long and a very short branch. And as you go up here, B gets larger and A gets larger with this axis. So if we look now and try and recover with different types of models and see how often do we get the right tree. If we use a single matrix model, we see the typical long branch attraction pattern in this top left-hand corner where the model's getting it wrong. Green means that it's getting the long branch attraction topology. If we use the mixture model, the correct model that it was simulated under, then we actually get perfect estimation for all regions of this, except for this top left-hand corner. There's slight less than perfect estimation there. Now for posterior mean frequency models, remember I said we had to use a guide tree to get those models in the first place. Well, PMSF1 is the case where the guide tree is the correct tree, and PMSF2 is the guide tree in the case that it's a long branch attraction tree. But even if we use those 2 different guide trees, we still get perfect estimation. So this means that even with the wrong guide tree, if you estimate site-specific frequencies using this method, you can now recover the correct tree. So that shows good performance. Now, if we do the same simulation under the long branch for Pell's setting, we can look up in the top left-hand corner. For LG plus F plus gamma, the simple single matrix model, it does very well. And there's a reason for that, and that's because this is a bias towards long branch attraction in the first place. So if you have long branches that really should go together, it's going to always get that correct. In the LG plus C20 plus F, the correct model, we see a slight long branch for Pell's bias in this top corner. So there's this pink means that the estimation was not perfect. For the PMSF1 model, the correct tree, we see quite a long branch for Pell's bias in the top left-hand corner, and same for when we use the LBR tree to do the fitting of the mixture model. So that's not so good for our model, for our PMSF models. However, the good news is that if you improve the taxonomic sampling, and I'll just go briefly through this, we look at this particular parameter setting here, and we now increase the taxon sampling to the 4 taxon tree problem, going from 4 to 20 taxa. The percentage of the correct tree with PMSF1 and PMSF2 actually goes from very low all the way up to 100% with 20 taxa. So improving taxonomic sampling drastically improves the ability to get the correct PMSF estimates and get the correct tree. So the long branch for Pell's problem goes away with taxon sampling. So finally, here's another simulation where I actually used a much more complicated mixture model, the C60 model, and then only recovered under the simpler models. And this is more like what reality is truly like. You have more complex data and your mixture model is simpler. And so if you do that under the long branch attracts scenario, LG plus F plus gamma gives a terrible long branch attraction problem. So does C20 model, the mixture model. Surprisingly, our PMSF1 actually does very well in that setting and gets everything right. PMS2 does not as well, but still better than the mixture model it was actually derived from and using the wrong tree. So that's an interesting kind of case of where the models seem to be doing slightly better than the actual mixture models. And if we look at the opposite long branch for Pell's setting, we actually get perfect estimation for all models except for PMSF2, where there's slight long branch for Pell's bias. So that's the simulation showing that the PMSF models actually do nearly as well and sometimes better than the mixture models from which they're derived. Finally, I'll finish with a real data example. And I realize I'm going over time here. Please, I'm sorry, bear with me. Here I have the case of the microsporidia. Now, microsporidia are a protist, a parasite, highly reduced intracellular parasite. And for many years, it was a mystery where these things fell in the phylogenetic tree of eukaryotes. It's become very clear in the last 2 decades that microsporidia are actually related to the fungi. This is a cross-section of a microsporidian spore in infected tissue. These are actually nasty parasites that people who are immune compromised can get. So microsporidia are extremely long branches in phylogenetic trees. And this is a very large concatenated data set by Brinkman et al. 2005. And this is showing microsporidia in the correct phylogenetic position. Now, if you analyze this data set with a single matrix model, what you get is a long branch attraction phenomenon that's very clear. This microsporidian long branch immediately joins the second longest branch in the tree, which is the branch that leads to the outgroup. Okay, so this is the so-called microsporidia plus archaea tree, and it's definitely not the correct tree. If you use the mixture model, such as the C20 mixture model, you get microsporidia going with fungi where they should. So the long branch attraction problem is dealt with effectively by the mixture model, the C20 mixture model. Now, if we use PMSF1, that is PMSF model derived using the correct tree as the guide tree, you see microsporidia going with fungi, and that's good. You get the right results. So our model is much better. Under PMSF2, where we used microsporidia going in the wrong place with the archaea as the guide tree, we still, when we estimate under that model, we will actually end up estimating the correct tree. So it's kind of robust to the guide tree that we gave it in the first place. So that's well behaved. And this is just an interesting histogram showing the differences in the likelihood between the correct tree and the incorrect tree. So anything positive on this diagram means the model prefers the microsporidia fungi tree. Anything negative means that the model favors the incorrect long branch attraction tree. And as you can see, the single matrix model doesn't do very well, likes the artifactual LBA tree. So does the simple mixture model, LG4X. The C4 model slightly does better, gets it right. And then as you improve your mixture models, you get well into the region of getting the right tree. And in fact, the PMSF models, based on the C20 mixture, are doing very well. Incidentally, if you partition this data set according to gene boundaries and allow for separate branch lengths, but don't take into account site heterogeneity, you don't deal with the long branch attraction problem, and you still estimate the wrong tree, despite quite a large improvement in model fit. So partitioning doesn't really work in this case to deal with the site heterogeneity induced long branch attraction problems. So I'll skip over the bootstrap results. We have other empirical examples where things, we can actually show that our PMSF models can overcome long branch attraction. There are two famous cases that Nicolas Lartillot examined in his paper with Hervé Philippe in 2007. One with nematodes branching either with arthropods or at the base of metazoa as the long branch attraction tree. In that case, our PMSF models get the correct tree, again, when the single matrix model doesn't. In the platyhelminths data set, where we expect, based on animal phylogeny, that platyhelminths should go with arthropods to the exclusion of deuterostomes, with the single matrix model, you get platyhelminths, again, going to the base of metazoa because of their long branch. But if you use the PMSF models, you can actually, if you use the sufficiently complex mixture, you can actually get the correct tree, just like cat GTR in the phylobase implementation does. But so this is a harder data set to get right. And if we use a complex enough mixture, the PMSF models can actually get the right tree, where the C60 model doesn't get the right tree. So there's some hope for our model. So in conclusion, I guess the overall conclusion is really that our models are idealizations, these so-called spherical cow models of the actual process of protein evolutions. The single matrix models are especially bad for a number of reasons. I mean, they've served their purpose, but there's a number of key model improvements that have been made in recent years that can actually help improve estimation. A frequent problem that occurs in both real data, well, in real data, is that when you get the model wrong, you will get systematic error, that is long branch attraction. And as you get more and more data, you will converge on the wrong answer. That is, the long branches going together. I hope I've showed that mixture models do seem to work well in this case to deal with especially site heterogeneity, which seems to be one of the biggest issues when it comes to large supermatrices and deep phylogenetic problems, is that site-specific heterogeneity must be correctly dealt with in order to actually estimate the correct tree. And our new PMSF models provide a viable alternative. And at this point, I should say that the newest versions of IQ-Tree, version 1.5 and higher, or 1.4.4 and higher, actually have this PMSF model implemented, and we have a manuscript in preparation. So with that, I'll conclude and say that that any questions are fair questions, and I hope I've made a convincing case.
Thank you very much, Andrew. That was the most clear explanation I've seen of rate variation and how it can get incorporated. So thank you very much for that. So I'd like to remind people that Google has removed the Q&A app, so you can't ask questions directly through, but you can tweet at Philobabble, or Phyloseminar. Tweet at Phyloseminar, or if you don't like using Twitter, there's a link on the Phyloseminar webpage in the attending tab that will take you to an IRC chat room where you could ask questions via chat. So I will just, hopefully people will figure that out. I will just ask a few questions here to get things going. You did a great job at the beginning of describing all the different ways in which the most common models are spherical cows. And then we spent the talk exploring one of those ways. Can you make a case of why rate variation might be the most important thing to look at? Or, I mean, I think that it shouldn't be a hard question for you, because I think that you're probably mostly interested in deep phylogeny and then somehow have ended up focusing on rate heterogeneity, and that wasn't by mistake. So. Yeah,
I can try to answer that. Should I keep it on screen sharing here, or is there?
Yeah.
Probably, huh? Yeah. Okay.
Maybe I have one other question that we'll ask specifically about a slide. So maybe just.
Okay.
Yeah.
Let me just say that I think, dealing with these deep phylogenetic problems, we've often tried, as these new different improvements in models have come out. So looking at compositional variation, looking at partitioning and allowing separate branch lengths for separate partitions in your data. We've actually developed quite a bit of models in the heterotachy area, looking at how rates of overall, site rates can shift over the tree. And I guess the subjective answer is that trying all of these things, the thing that tends to make the biggest difference over and over when we do analyses is looking at site-to-site heterogeneity in the site, basically site profiles. So I think this issue of having a restricted alphabet of amino acids effectively at every site, when the single matrix models are assuming that all 20 amino acids are accessible, that's a big problem. And it's sort of like doing a Jukes-Cantor distance correction under assuming 20 states, when in fact you really should be using one with 4 states. You're going to get an underestimation of the longer distances, and that's always going to lead to this long branch attraction problem. So that's, I guess, that's the argument I would make in that case.
Yeah, with that specific issue, going back to Richard Goldstein's talk again, I mean, he did make even just based on simulation in terms of protein stability, he made a pretty convincing case that at any given time there's only a few amino acids that should actually be accessible from a given state.
Mm-hmm. Yeah. And I think that's the crux of the issue, is that if you don't take that into account, you're not going to get it right with long branches. You won't get the number of substitutions right on the longest branches. And that'll lead to systematic bias in which tree is most likely.
This is sort of a hard problem on top of a really hard problem, namely timing on top of deep divergences. But so far, you sort of focus mostly on the topological aspect. So, imagine that these models make a huge difference for branch lengths as well. I mean, do you guys ever try to use branch lengths to say something about the relative timing events or something like that? Or is that just sort of viewed as too hard?
we actually have, we've looked at that issue, especially in the case of trying to figure out what the age of the major eukaryote clades are, these so-called supergroups. We've been using some of the relaxed clock models that are implemented in Phylobase and comparing what you get if you use single matrix models versus these site heterogeneous models. There is an effect. So there are different, you do get different ages. The problem is that, of course, it's confounded with the issue of which is the right relaxed molecular clock model to be using, and also what kinds of, how to deal with fossil constraints and what kinds of bounds you need to put on fossil constraints. And so we actually have a paper in a Cold Spring Harbor perspectives volume where we actually explore all of those issues. And the answer, I guess the short answer is it's really complicated, but yeah, the site heterogeneity does make a difference to molecular clock analyses.
Would you mind going back to the slide where you introduced the PMSF models? I don't feel like I totally get it.
Okay, so yeah, I kind of realized I was running out of time at that point. So with the mixture model, we're going to evaluate the mixture at every site, and then basically we're going to be doing this to get the site likelihoods for each class. Okay, and so what we're doing here with the posterior mean frequency approximation is basically, like I said, empirical based. We try to find the posterior probability of a given class, given the data, using this empirical Bayes type equation. And then we have two choices. We actually have tried several options here. One is we could just assign that class to a site and say, okay, this is going to be a hard-coded class to that site, and we're going to evaluate the likelihood under that class only for the site. We have done that, and it actually turns out to not perform very well, or as well, as what we do now, which is a sort of this conditional expectation expression here instead. So instead of the conditional mode estimator, we're doing a conditional mean estimate of the site frequencies. So multiply the frequency of a given amino acid times its posterior probability, sum it up over all the classes to get the kind of averaged frequency for that site. And so then if you have two classes that don't fit the site very well, so imagine that the real distribution of site class is much richer than our mixture. We may not find that any of the classes fit that well, but there will be a few that fit okay. And if you can actually have those, say they have the same posterior probability, you can actually factor that in and somehow get an averaged estimate of the frequency that's somewhere in between those two not very probable classes. So it's almost like a smoothing procedure for a discrete, we've got this discrete distribution approximating something that's very, very rich. And this posterior mean approach tries to get you somewhere in between some of the classes to more smoothly fit the real data at that site without actually directly estimating a free parameter at that site.
Okay, that's really helpful. So, I mean, basically, yeah. So you do have to sort of exponentiate a different rate matrix at every site, but it's still
right.
sort of one likelihood calculation.
That's right. And that's what leads to the slightly greater computational cost over the regular single matrix model.
And so can you say, like, I mean, given that you have a model like this, it seems like the the role of the per class models is somewhat different. I mean, you could even start with sort of some stupid per class models, and then the fact that you're estimating a correct posterior mean of them per site would sort of make up for that. I mean, so what are the models that you start with in this?
You can start with anything. I mean, we generally use the C20 or C60 mixture that Le Gascuol and Lartiot came up with. So anywhere from 20 classes to 60 classes that they estimated from a database a number of years ago. And from those, I mean, ideally we use the richer mixture model, but then we have to fit the mixture model on a guide tree to begin with, which has all the RAM costs of the original mixture model. So that actually is currently a slight limitation to this approach, is that if you want to use a really good mixture model to get these PMSFs, you're going to be stuck with needing nodes with 500 gigabytes of RAM on them. Although Min has come up with some ideas about economizing on the RAM for this particular phase of the calculation.
I mean, what I'm kind of trying to get at is that, like, it's almost as if you sort of want these submodels, these per class models. They don't need to be good models in themselves. So you could imagine them being almost like principal components. I mean, maybe that's already, if you are estimating in a mixture model framework, maybe that's already sort of built in. But I mean, you could have, for instance, some really, really good models in the context of when they are getting mixed, but each individual model could be sort of horrible.
Mm-hmm. Yeah, but so I mean, so you're thinking that it's just a matter of if you make the mixture complex enough, you might be able to approximate sort of any frequency distribution with this kind of an approach.
Yeah, maybe, but yeah, maybe. But I mean, yeah, I mean, you could come up with a good set of principal components that might not require so many components. I mean, there was a paper by, I think, some Swiss fellows who were doing like principal components in the space of protein substitution matrices.
Oh yeah, yeah. I think. Well, I mean, there was Bastien Bousseau and Matthew Gosselin, I think, also had some approaches that were dealing with compositional heterogeneity that dealt with that principal components type approach.
Yeah, well, I'm not going to be able to pull it up. But anyway, that's yeah. Of course, principal components shows up a lot in my database of papers. Oh, here it is. Zoller and Schneider, improving phylogenetic inference with a semi-empirical substitution model. Anyway, I've sort of rambled on for long enough.
Mm-hmm.
So it looks like, yeah, either people are feeling quiet or it's been a little bit too confusing about how to ask questions. If you have any feedback about how to ask questions or like a better way of doing this, just drop me an email or send a tweet to Phyloseminar. So I guess maybe, Andrew, you should finally unshare your screen.
Okay. I'll just leave that there. In case anyone wants to ask questions, there's emails there as well.
Right, okay.
Well, thank you.
Well, thank you very much. That was, like I say, an excellent seminar, and I guess we will look forward to the next one, which will provide a slightly different perspective on how we can model substitution processes. So thanks again, Andrew, and yes, see everyone in about a month.
Great, thank you.
See ya.